Megha is a PhD student in the Omar Ahmed lab. I think, Megha, your original background is in engineering, electrical engineering. Am I remembering this correctly?
Right. I did a master's in electrical engineering at the University of Michigan.
Right. So I think, but has had the good judgment to migrate into the world of neuroscience. So she's going to talk about some very interesting work that's been going on in terms of the retrosplenial cortex. So please go ahead.
Okay. Thank you. And I'm really thankful and excited for this opportunity to tell you all about the rhythms of the retrosplenial cortex during movement and REM sleep. But before we go into all the details, what is the retrosplenial cortex? The retrosplenial cortex is a midline structure located just posterior to the splenium of the corpus callosum, and it's conserved across species. In rodents, it spans about 6 millimeters rostrocaudally. But why study the retrosplenial cortex in Parkinson's disease? So some of the symptoms of Parkinson's are bradykinesia, freezing of gait, REM behavioral disorders, facial disorientation, and amnesia. While some of the symptoms of damage to the retrosplenial cortex are spatial disorientation and amnesia. In addition, altered retrosplenial activity is correlated with freezing of gait. So we can see that there is considerable overlap between symptoms of Parkinson's disease and changes to the retrosplenial cortex. So it's important to study the retrosplenial cortex in Parkinson's disease. And in addition, there is cholinergic dysfunction, which happens in Parkinson's disease, which affects all of these symptoms. So we're interested to know that what impact the impaired cholinergic inputs to the retrosplenial cortex has, and in order to understand this, we need to know how the retrosplenial cortex codes information during normal cholinergic functioning. So we see increased activity of cholinergic neurons during REM sleep and active movement in both basal cholinergic forebrain cells, basal forebrain cholinergic cells, and the brainstem cholinergic cells. This is the reason why we focused all of our electrophysiological recordings from the retrosplenial cortex during REM sleep and awake active states in rodents. But before we go into all the details of the recordings, let's understand what we would expect to see when we have an electrode inside the brain in the middle of a bunch of neurons. We would get a time-varying voltage signal, which are called local field potentials. But what do these mean? So consider for an instance, like if you can record individually from each of the neurons surrounding an electrode, and you measure the electrical activity of each of these neurons. So in that case, the summed activity of these cells would be represented by the local field potentials. One interesting point to note here is that the electrical activity of each of these neurons is different, and the lack of any synchrony among their activity is manifested in the lack of a definite rhythm in these extracellular potentials. So we see that random activity of neurons gives rise to a random local field potential. But what happens when the activity of these neurons is synchronized? For instance, say the peaks and troughs of the electrical activity across neurons is perfectly aligned. In that case, we would get a high amplitude rhythmic signal. So since oscillations represent neuronal synchrony, they allow studying the dynamics of an entire network without studying every single neuron of the network. So one important property of neural oscillations, which we deal with, is known as frequency. And that can be seen as the distance between adjacent peaks or troughs, which gives us the time period, and the inverse of that is the frequency. So frequency gives us the number of cycles that happen in 1 second. So now that we understand what local field potentials are and what oscillations are, let's look at the oscillations found in the retrospinal cortex during REM sleep and active behavior. So the dominant oscillations in the rodent brain during both REM sleep and active behavior are these 8 Hz theta rhythms seen here in the local field potential. Another way to visualize this would be in a power spectrum, which gives us the distribution of power across frequencies. And in this plot, we see increased theta power in both CA1 and V1, which is seen during both REM sleep and active behavior. Now, these theta oscillations are also seen in the retrospinal cortex during both of these brain states. In addition to theta oscillations, we see another oscillation found in the hippocampus and other spatial navigation regions, specifically again during REM sleep and active movement, which are known as gamma rhythms. These are these 30 to 80 Hertz oscillations, as can be seen here in the bump in the power spectrum. So gamma rhythms are also seen in the retrospinal cortex. So we see theta, we see gamma in both retrospinal cortex and other brain regions involved in spatial navigation. But in addition, there is another unique, much faster oscillation seen specifically in the retrospinal cortex. So again, you can see a clear bump in the power spectrum at around 140 Hertz, which is specifically happening only in the retrospinal cortex. Feel free to interrupt me at any point if you have questions, by the way. I would love for it to be interactive. Okay, so now that we see this interesting, unique oscillation, which is centered at around 140 Hertz, specifically in the retrospinal cortex, we look further at its properties. And we see that these 140 Hertz oscillations are strongly coupled to the peak of the slower theta rhythms. If we zoom in further into a single theta cycle, we can see how precisely these 140 Hertz oscillations are coupled to the peak of theta. To quantify this strength of coupling, we assigned a phase to each point on the theta cycle such that the peak is represented by 180 degrees. So averaged across 9 rats, thousands of theta cycles, we see how precisely these oscillations are coupled to the peak of theta. So these fast rhythms resemble the teeth or ridges on a gear, which are called splines, and hence we call these fast oscillations as splines. So now that I've introduced you to retrospinal splines, let's look at their interesting properties. So splines demarcate high activity REM frames. And here you can see that in a single REM epoch, splines occur in clusters denoted by these dotted gray lines. And these clusters are correlated with increased firing rate of both fast spiking and regular spiking cells. Further, we saw that retrospinal cortex spans about 6 millimeters in the rat brain. And so we saw that the splines are correlated across the entire long axis of the retrospinal cortex. But the correlation with gamma in both retrospinal cortex and hippocampus is low. So given this interesting correlation across the entire long axis, we wanted to know if the strength of splines is uniform across all the layers, or is there a specific gradient in power? So for this, we performed bilateral recordings in the retrospinal cortex, and we found that both splines and gamma are strongest in the superficial layers, and their power decreases with increasing distance from the midline. And this holds true for both REM sleep and freely moving behavior. So given the presence of this unique oscillation, we wanted to know if they're involved in long distance communication, and specifically during interhemispheric communication, which is important for attentional motor integration. So we asked if gamma rhythms and splines in the retrospinal cortex are involved in interhemispheric communication. And we saw something very interesting. So we found that splines are anti-phase across hemispheres, such that the peak of splines in one hemisphere corresponds to the trough in the other hemisphere. And you can see consistently all the cycles of splines that we see locked to the peak of theta are consistently anti-phase across hemispheres. However, gamma rhythms are perfectly in phase, with their peaks and troughs perfectly aligned. Again, to quantify this phase relationship, we plotted the phase offset of both of these oscillations across hemispheres and found that splines are consistently anti-phase, denoted by a phase offset of 180 degrees, while gamma oscillations are perfectly in phase. Again, we saw this in the entire population, that gamma rhythms are perfectly in phase, both during REM sleep and freely moving behavior, while splines are perfectly anti-phase. So we find that this phase relationship is strong in REM sleep and splines are very strong in REM sleep. But further, if we were to divide REM sleep between tonic and phasic REM, do we see any differences? So as expected, we see an increased power in both splines and gamma band during phasic REM. However, the phase offset is consistent across both of these REM states, with splines being perfectly anti-phase and gamma oscillations being perfectly in phase during both tonic and phasic REM.
Yeah. So Megha, could you say a couple words about what's the differentiation between, for those of us who are not sleep specialists, tonic and phasic REM?
So phasic REM is seen with increased muscle twitches and increased these PGO waves which are seen in the brainstem, which correspond to increased activation during one subset of REM epoch, which is known as phasic. So that is manifested with increased power in both theta and higher frequencies. So behaviorally, you see more activation in terms of twitches and rapid eye movement. And in terms of electrophysiological signatures, you see increased power in both theta rhythms and these fast oscillations. Okay. So now that we have looked at REM sleep and broadly at the freely moving active states, we wanted to know further if movement speed specifically affects splines and this interhemispheric communication. So we looked at rats running on a linear track, and we found that both splines and gamma have increased power with increasing running speed. Again, we saw this in mice as well, which were head fixed and running on a ball. As speed increases, the coupling of both splines and gamma increases with their movement speed. But what about the interhemispheric communication? Both spline and interhemispheric coherence increases with increasing movement speed. However, the precision of this phase offset increases precisely for splines as compared to gamma at higher running speeds. So we see this interesting oscillation, which has a unique interhemispheric phase relationship. But since oscillations represent synchronous neuronal activity, do retrosplenal units show phase locking to splines? So here is an example of raw traces from both hemispheres, again showing perfectly anti-phase splines, with spikes happening at the trough of splines in the ipsilateral hemisphere, which are aligned to the peak of splines in the contralateral hemispheres. To quantify this phase locking relationship, we did something similar as we saw with theta oscillations. We assign a phase to every point on this spline cycle, and then we look at the distribution of those phases. And we see that the preferred phase distribution shows clear phase locking of spikes to the trough of splines in the ipsilateral hemisphere, but the peak of splines in the contralateral hemisphere. And this is seen in both single units and multi-units. The precise phase locking of spikes is to opposite phases across hemispheres. So to summarize, I would like to say that unlike other brain regions, the retrosplenial cortex has two distinct channels of rapid interhemispheric communication, perfectly synchronous gamma and perfectly anti-phase splines. And this interhemispheric communication mediated by splines becomes more precise with increasing movement speed. And cells in the retrosplenial cortex phase lock to the trough of ipsilateral splines, but the peak of contralateral splines. So now that we have some information about the activity in the retrosplenial cortex during cholinergic brain states, what next? So we want to understand how cholinergic dysfunction affects both splines and gamma across retrosplenial layers, and how the interhemispheric communication in both of these frequency ranges is affected during REM sleep and an escape to shelter task. So this would give us an idea about the mechanisms of splines and the role of cholinergic inputs in both of these oscillations. With this, I would like to end my talk, and I would like to thank the entire Omar lab and all our funding sources. Thank you. I'd love to have more questions.
So we're, oh, Fimo?
Yes, yeah, hi, yeah, that was really nice talk. So I'm interested in your splines. You mentioned compared to the CA1 region, and one of the other reasons you looked at, it seems to be unique to this retrosplenial cortex. Did I say it right?
Correct.
But did you, how much mapping did you do across the entire cortex, cerebral cortex? I mean, if you look at a visual cortex, parietal cortex, and frontal cortex, and so forth. So how confident are you to saying that it's unique to the retrosplenial cortex?
Yeah, I did not have that slide. So we looked at the hippocampus, at visual cortex, posterior parietal cortex, and retrosplenial cortex. And we found that across all of these regions, splines are strongest in the retrosplenial cortex specifically. So,
but if you look, did you look at other cortical regions?
No, these are the 4 brain regions that we study. But so the literature says that these higher frequency oscillation are found in the hippocampus, the posterior parietal cortex, and somatosensory cortex. But so among all of these regions, we studied 3 of these regions and found that splines are strongest in the retrosplenial cortex. So yes, we would love to study other brain regions as well and study if those fast oscillations, which are mentioned in the somatosensory cortex, have similar properties as we see in splines, or are they different? And if they are stronger in the retrosplenial cortex as compared to the somatosensory cortex.
Okay, so my lab studied sleep also using EEG, not local field potentials, but when we map the REM sleep electrical activities, that high gamma phase coupling with theta seems to be quite widespread. It is definitely not restricted to just retrosplenial cortex. And the frontal cortex is a lot much weaker, but parietal cortex and visual cortex and seems to be quite wide. So that's one thing I wanted to come. I don't know whether there's a difference between the mice and rats. So the other question that I have, so that's one comment that I have. The other question that I have is that in terms of the anti-phase behavior, this is something we saw also when we looked at cortical cardiac coupling in the dying rats. And in fact, the left side and the right side shows the anti-phase behavior. When in the dying animals, there's a surge of a cortical cardiac coupling. And the electrical coupling, the left side and right side is opposite phase. So I'm wondering whether there's a common mechanism behind these two types of electrical activities. It's kind of
Which
curious.
frequency were you looking at for that?
Oh, this is we're looking at, okay, so I think this so we're looking at multiple frequencies that I can chat with you afterwards about that.
Okay. Yeah, that's very interesting. I did not know about that, but that's very interesting to see if that's similar to what we see.
Yeah, but ours is, of course, it's not restricted to REM sleep or not even during sleep. This is the animal is comatose and dying. So, and it's a unique form of coupling that it doesn't exist in normal animals. Yeah.
Anyway. Okay, thank you for that. I will follow that up.
Sure.
So, question about the difference between the tonic and the phasic states when you do see some modulation of these effects. So what do you think might be driving that? I mean, the implications, you think there's some subcortical inputs that are modulating that effect, but do you think that, do you have any specific hypothesis for that, or?
I would expect the brainstem cholinergic cells to be driving it. But again, to have a definite answer, we would have to record simultaneously from both of these regions, from retrosplenial cortex and the brainstem regions to understand that.
Yeah. Okay. Okay. Any other questions or comments for Megha? Okay. I think in that case we're gonna conclude a little early today. Thanks for a really nice talk, and we will be I think meeting again in a month's time. And I think Nancy, there's an executive committee meeting after that next month's meeting as well. Is that correct?
That's correct.
Okay. So we'll have another, see everybody in about a month's time. And so this give you back a little bit of extra time today to catch up on all the stuff, at least for me, all the stuff I should have done over the holidays. So all right. Thanks very much, Megha.